Hi everybody and thanks for watching. If you are watching the other talks in the session, you will notice that this talk ends the spongy part of the session in which we heard some great talks regarding various aspects of sponge ecophysiology. Clearly, water pumping is a key parameter in sponge physiology, and our goal in this short presentation is not to present new findings or provide an extensive review of the topic. Instead, we aim to highlight some unresolved puzzles and knowledge gaps regarding the factors that control sponge pumping. Some are hunting the field for many decades. Our hope is that delineating the open questions along with some speculations about the potential mechanism will entice some of you to literally jump into the water and tackle these exciting questions in the coming years. Just a quick reminder that sponges are very simple organisms that lack real tissue and organs and have no equivalent to the nervous system of most other metazoans. Moreover, despite many decades of searching, no alternative electrical conduction system was located. Best to understand what Sally is telling me, we now know that there are exciting evidence that demosponges may be using chemical cues to conduct signals through the sponge body. This is done in a similar fashion to the way most metazoans are using hormones and neurotransmitters, but the rate and the extent that these chemicals are working is yet to be seen. Glass sponges are an exception to this rule, as the entire sponge body is multinucleated syncytium, for which calcium-based action potential can propagate slowly and halts the flagella action in an all or none fashion. This ability allows glass sponges to arrest their pumping almost instantly, as in this example here. See the record of pumping rate stops almost immediately. This is a kind of behavior we call the rests. And this allows the sponge to respond, for example, quickly to event like sediment resuspension, such as in the experiment shown here. Also important to recall that flow of water for the sponge is unidirectional and very fast, meaning that using flowing water to transport chemical signaling is not an efficient or actually feasible way to communicate within the sponge. In his seminal work of the pumping rate of Caribbean sponges, Henry Ryswick noticed that some sponges pumps continuously for a prolonged period of time and responded only passively to elevated level of suspended sediment during storms, for example. Other sponges, such as Verongia, maintained high level of pumping, as noted in these plots from Henry's thermistor, the paper plots made over 60 years ago. But this pumping activity lasted a long period, ranging from 6 to 40 hours. But they were interrupted by short periods of complete cessation, and interestingly, these cessations shown in this example as red arrows were asynchronous throughout the population. So Ryswick suggested that they were inherent in origin. The reduction of flow was associated with contraction of the atrial lining. You can see here in this is a picture of an actively pumping sponge, and this is a picture of the same atrium when the sponge stops pumping. And this contraction included the exhalent canals and their apertures as shown in this lower picture. Ryswick also noted that the process was slower for larger specimens, and he attributed this difference to the slow spread of contraction along the exhalent system from unidentified site of initiation. For the shallow water sponge Henry called Tetiacrypta, he noticed that a very complex activity pattern. All active sponges exhibited a clear daily cycle of contraction during nighttime and expansion during daytime, and this contraction expansion cycle was accompanied by variation of the pumping rates that are plotted here. Over a longer time span, each individual maintained fairly uniform cyclic period ranging of expansion and contraction. And this time period ranged from 9 to 21 days and were asynchronous throughout the population during the calm season. Rasbig suggested that changes of T-crypt activity were probably due to changes in the flagellar activity because he couldn't imagine how flagella will keep working for days in a non-pumping sponge. But I don't think he had any data to support this hypothesis. I should note that it is high time that 60 years later, this remarkable work will be repeated with modern tools. More recent observation of in situ pumping in glass sponges revealed that the flow rate out of the oscula of two sponges, recorded with an acoustic current meter and plotted here in red and blue, was tightly coupled with the ambient current speed, plotted in green. But as discussed by Evgeny Matayev earlier in this session, it seems that this flow is not passive, but rather is tightly controlled by the sponge. See, for example, the increase of flow of the ambient flow that is not accompanied by a similar increase of the sponge from the flow. And for these reasons, it is suggested that to call this type of behavior a sense-induced flow. As we know, the pumping rate of most demosponges is uncoupled to the ambient flow. Here is an example from the coral reef sponge Tionella swinoi. The X current speed recorded with this acoustic current meter is plotted in red, and most of the time it is actually much faster than the ambient flow speed recorded by this current meter and plotted in blue. And the two are totally unrelated. Interestingly, there are periods where the sponge reduced dramatically its pumping rate, sometimes close to zero, with no visible triggers that we could locate. In this case, recorded from a different specimen, not the one that is in the picture, we were lucky. We noticed that the pumping stops every evening at exactly the same hour, just after sunset. Luckily, we had a video camera installed to monitor the system. Examining the video, we noticed that a sea urchin would go out in that time of the day, and when it crawled over the sponge would stop pumping. Who would imagine? To compare the pumping behavior of adjacent sponge under controlled condition, we set up an automatic dye tracking system that measured the X current speed of two sponges positioned next to each other in a well-mixed water table, meaning that both sponges experience exactly the same conditions, and every half an hour the flow from two adjacent oscula in the two adjacent sponges would be measured. Plotting with flow rates one against the other, you can see surprisingly that the flow patterns were completely independent of each other. So again, maybe suggesting inherent control. While in some sponges the oscula diameter changed very little, in others the oscula is very dynamic. Concurrent recording of oscula area, so this area, plotted here in yellow, and the X current jet speed, the ES, plotted in black, from three different oscula of the sponge Cliona orientalis, indicate that in most cases pumping rate increased with the opening of the oscula. Note that if the pumping rate was controlled by the osculum size, the ES should have increased when the oscula constricts. Indeed, in some cases, the oscula size decreases while pumping rate remains high, suggesting that the control of the pumping rate is partly independent from the oscula dynamics. In these videos, you can see side view and top view of the osculum of a very small juvenile of the freshwater sponge Ephidetia. And when I'll start the video for you, note the osculum here in the side view first, see how it inflates, and actually when the oscula closes, the entire sponge body, I'll play it again, the entire sponge body inflate, and then when the oscula opened again, it deflates. Now, more interesting even is to watch it from top. Look how the canal system is changing its dimension during this sneezing of the sponge. I'll play it again so you can see. Watch the canal system, the aquiferous system, how it changed its dimension. We don't know if such changes occur in adult sponges. Finally, recall that we now know that sponges possess flow-sensing cilia that are distributed within the sponge oscula and potentially also throughout its aquiferous system. This is again the side view on the top view of a Fidetia. And if you look into the osculum using electron microscopy, you can see the small cilia that extend from each cell that lines the inner part of the oscula. And these rigid cilia are now considered to be flow sensors. So, this short review of some of the documented dynamic of sponge pumping leave us with more questions than answers. Some are mechanistic in nature. For example, can demosponges halt their flagella bit? And can they modulate the flagella bit rate? What is the role of mucus in the sponge filter? We know from the work presented earlier in this session by Saeed Asadzadeh that CFD modeling suggests that at least some segments of the collar should be sealed, and there are evidence that this seal is made of mucus, or it is mucoid in nature. We also ask to what extent do adult sponges dilate and constrict the canals of the aquiferous system in a similar fashion to what we have seen for juveniles and explants? And where are the valves and sphincters located? And how are these related to the location of flow sensing cilia? Another unresolved question is the nature of the sponge functional pumping unit. Are they the guanocytes, the chamber, or the aquiferous system? On a more fundamental level, we ask what is the mode of coordination in sponges? Here, we would like to put forward a proposition. It may be time, after almost 150 years of research, to abandon the search for conduction systems that allow communication between the various parts of the sponge and facilitate central or maybe distributed control, as is the case in other metazoans. Instead, we suggest to consider the alternative, that the ability of sponge to behave, control their pumping, and react to environmental cues may be a consequence of swarm behavior of independent and disconnected functional or operational units rather than a coordinated action. I am no expert in the field, but when you read about the subject, you find definitions for swarm behavior such as the collective motion of a large number of self-propelled entities, or sometimes it is also described as an emergent behavior arising from simple rules that are followed by individuals and does not involve any central coordination. So it seems fit to me. To give you an example, consider this simple experiment with Bristol boats. These are essentially toothbrush heads with a motor installed on top. There are no sensors, no CPUs, and no way to communicate between each other. All they do is move around in a certain motion, and they can be programmed to move in different motions. And note that when the number of units is small, they move in a random fashion. But when they reach a critical mass, interesting patterns appears. The boats are organized in order clusters that move around the edge of the arena, displaying an apparent organized behavior pattern that is both predictable and repeated. I think we should also have a side view of the boats. Let's give it one more second. Here you can see them from the side. And as I said, you can program them to spin or to walk, and they always create these nicely organized clusters. Therefore, we propose that perhaps a useful approach would be to consider sponge as a smart decentralized system. That this approach will provide us with a simple explanation to numerous observations. For example, the erratic pumping patterns with unexplained fluctuation of the pumping rate may be simply a consequence of the independent activity patterns of the sponge functional units. Similarly, accepting the idea that there is null or maybe minimal communication between the sponge functional units, the mechanisms underlying phenomena like the lack of synchronization between different oscula of the same sponge or between different sponges in the population is trivial. Some of the behavioral patterns we conceived as coordinated action can be simply explained as a result of common response of independent functional units to the same trigger. For example, clogging of the aquiferous system due to sediment resuspension. So, there is a lot to think about and a lot of work in front of us. And thanks for listening. I hope you will enjoy the rest of the session. I just want to end by thanking all our friends and colleagues for many hours of interesting and fertile discussions, and to all our students that challenge us on a daily basis. Keep asking questions. Thanks.